अस्सलामुअलैकुम वसीम। जी, थैंक यू। आगे हैं सर। थैंक यू। अस्सलामुअलैकुम सर।
Assalamualaikum, brother, sister Khatija, how are you?
I'm fine, thank you, Prof. Iqbal.
Good to see you. Thank you for accepting our invitation. Good to see you after a long time.
Well, I was in Karachi earlier this year.
Yeah, you should come back. I think you should come more often.
Yeah.
Inshallah. Okay, Khazima, we have someone of a very high quality and which we are so proud of her achievement, mashallah.
All right, sir. Sir, with your permission, can we start?
Yes.
Thank you very much. Bismillahirrahmanirrahim. Assalamu alaikum, ladies and gentlemen. I'm Hazima Mozam, and I welcome you all to Comstech Distinguished Scholars webinar series. Today's webinar will be delivered by Professor Dr. Khadija Muhammad Yusuf. Dr. Khadija is currently the Vice President of Islamic World Academy of Sciences, and she is associated with National Institutes of Biotechnology based in Malaysia. The title of our talk today is Transforming Newcastle Disease Virus from Menace to Potential Therapeutic Ally. With this, on behalf of Comstech and myself, I would like to thank Professor Dr. Khadija for her time and for accepting our invitation. To formally introduce and welcome our speaker, I would request His Excellency, Professor Dr. Muhammad Iqbal Chaudhry, to welcome and introduce our guest speaker today. Over to you, sir.
Bismillahirrahmanirrahim. Assalamu alaikum. Good morning, good afternoon, good evening, ladies and gentlemen. We already have a full forum. I think there's over 80 people already online, which not only reflect the interest in topic, but also the stature of very high quality scientists. Professor Khatija, our very distinguished speaker, she is an academician of Islamic Academy of Sciences and also the vice president. So we have had the honor of hosting her, though briefly, in Karachi during the IAS conference at the International Center for Chemical and Biological Sciences. She is a very prominent researcher in the field of biological sciences, particularly in the field of virology, and she leads this very prestigious institution called National Institute of Biotechnology, Malaysia. Professor Khadija Muhammad Yusuf has extensive engagement and networking with industries, professional bodies, and academies. She is currently the Vice President of Islamic World Academy of Sciences, member of the Council of Scientific Advisors of International Center for Genetic Engineering and Biotechnology, ICGEB, and ICGEB is a partner of Comstech in many YC-related initiatives. She's a senior fellow of the Academy of Sciences of Malaysia, a fellow of the Malaysian Scientific Association, and fellow and former president of the World Academy of Sciences for the advancement of science in developing countries, TWAS. Her 5-year term as the Deputy Secretary General of the Ministry of Science, Technology and Innovation, Malaysia, has been a great time in which she has actually promoted science and technology through national policies and developing framework in managing sciences in the country. And I'm sure that her experience in science management as well as national policy framework is equally important as her experience in the field of science. She is a strong believer in the need for translating science into tangible benefit to the people and for the benefit of common good. She sat on board of trustees of the International Livestock Research Institute and governing body advisory board of Lathrop Asia and technical advisory panel for ComSets. as well as several agencies in Malaysia. She's currently the chairman of the National Committee on Research Integrity. Her research focus on molecular biology of Newcastle disease virus, which is a very, very important, economically important virus in terms of its impact on a range of different diseases, particularly in poultry. Through reverse genetics, she's currently developing an NDV-based cancer vaccine using NDV virus as a carrier for vaccine development and for the treatment of colorectal and bladder cancer. Her research innovation have received great recognition nationally and internationally, such as the prestigious Medica Award and UNESCO Carlos Finlay Prize for Microbiology, which both are very prestigious honors. She was recognized as one of the top 20 most influential women in science in the Islamic world. She was also featured in 2021 special edition of DC Comics Wonder Woman, Wonderful Woman of the World. Her special message to young scientists looking to impact their field is to never give up and value the importance of teamwork. As we and our Muslim Ummah looks for role models, I'm sure Dr. Khatija is someone to look towards and to learn and to be inspired. With this, I'd like to invite Sister Professor Dr. Khatija Muhammad Yusuf for her presentation. Professor Khadija.
Thank you, Prof. Iqbal. Members of Comstech, ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh. I'm not sure, it's probably good afternoon over there, but it's already evening and nearly Maghrib over here. Assalamualaikum and Bismillahirrahmanirrahim. The title of my talk today is Transforming Newcastle Disease Virus from Menace to Potential Therapeutic Ally. The picture on this slide, you all can hear me, right?
Yes, we can hear you and we can see your slide as well. Thank you.
Yeah, the picture of this slide is where my office is. We are at the Malaysia Genome and Vaccine Institute. Now, how do I, I'm part of the National Institutes of Biotechnology Malaysia, and we have 3 institutes there under NIBM. 2 are located in the Klang Valley, where Kuala Lumpur is, and 1 is located in the north, and that is the Penang. So we have the Malaysia Genome and Vaccine Institute, MGVI. We have the National Institute of Pharmaceutical and Nutraceuticals, IPharm in Penang. And we have the Agro Biotechnology Institute in Kuala Lumpur. I hope that we can work together and do some collaboration. The core facilities that we have in the institute that I'm working in, we have sequencing, we have mass spec, but not as many as ICCBS. We have Genome Computing Center, Protein Engineering and Bioprocess, Genomics, and we do structural biology, but I do on vaccine research. So let me introduce you to Newcastle disease virus. It is a member of the Paramyxoviridae, and is an orthoavular virus, avian orthoavular 1. It was first discovered and isolated at Newcastle upon Tyne, and also from Indonesia in 1926. It is spread through infected birds, droppings, secretions, and it can be spread rapidly amongst the confined birds. Now, NDV affects more than 241 species of birds. It is a respiratory disease, it's prevalent worldwide and endemic in many countries in Asia, Africa, and the Middle East. So Why is it endemic? Because of inadequate vaccination, interference by maternal antibodies, poor administration of vaccine, concurrent infection and immunosuppression. And then we have also these wild migratory birds, which are reservoirs for the virulent virus. So these birds migrate from north to south and south to north annually, and they will spread the virus. We also have presence of antigenic variants of NDV strains. So these are some of the clinical signs of the virus. I won't go through that. And we have also the poultry industry. It affects, and we have poultry meat, and there are more than 130 million metric tons produced every year. Now, the virus is an autoavilovirus 1. It's got this range of serotypes. I think I have got the wrong set of slides. Never mind, we'll go on. So it's got a wide host range. The virus structure is pleomorphic. Yeah.
Khadija, I think take the time and change it. I mean, we are
okay.
all
Yeah, because I'll do that. Hold on. Yeah.
We would like to hear a lot about this very important topic, please. So
yeah, just a minute. I can't remember. Yeah, this is the one. Can you see this?
not yet.
Hold on. Hold on, yeah.
Oh, sure.
No, this is not the one, sorry. Just a minute. Comstech.
Professor Khadija, the slides that you sent to me
Yeah.
in the morning, should I play them? Let me see.
I've got it now.
Okay.
You see, I always have this strong belief in Murphy's Law. and you never know what. Can you see my slides now, or I have got to go out?
No, we cannot see your slides.
No,
you can't see, right?
No, not yet. I think what
okay,
you
hold
can.
on. I'll do this.
Dr. Khadija, I would suggest that if you log out and log in again, it will be much easier for you, just to avoid any extra technical steps.
Is it? Well, the thing is, I think I've got it now. Just a minute. Can you see now?
Yeah. Yes.
Okay. Yeah, sorry about that. I was rushing from another meeting. So it is endemic in many countries in Asia, Africa, and Middle East, but and there are frequent outbreaks of the disease. The top producers are Brazil, USA, and China for poultry. Hold on. Now, the NDV structure, the virus is pleomorphic, is about 100 to 500 nanometers in diameter. It's an envelope virus with a helical nucleocapsid. It's got F and HN glycoproteins on the outside, and then the matrix protein, the nucleocapsid protein, the P phosphoprotein, and the large polymerase protein. The NP, P, and L proteins make up the polymerase and transcriptive complex. Now, the virus is a single-stranded negative sense RNA genome, and the various genes that encode for the various proteins are like, as you see here, from 3' to 5', NPP,
Dr. Hari, I'm sorry to interrupt, but we cannot see your slides.
You can see?
No. Let me just try to share it from my side if these are the correct slides that you have shared today.
Okay.
Can you see my slide?
Wait.
Screen?
Yeah. Hold on. Okay, I'll use your slides.
Okay,
control
just
it.
let me know when to move forward.
Okay, next.
Okay.
Next. Now before that. The virus replicates according to the rule of 6, that is, it needs 6 nucleotides for it to be an active virus, okay? The genome has got 3 different sizes of genome, from 15,186 kilobases to 15,198 bases. Next. The virus that I've been working with is called NDV strain AF2240. It's a local Malaysian isolate. This virus is heat resistant. It's very, very virulent. You can see from the picture here that it kills nearly 100% mortality. It belongs to the genotype 8. Next slide. Now, the virus itself has many genotypes. And we found that one of the reasons why we have vaccination failure is because there is a genotype mismatch between the circulating Malaysian field strains, is the genotype 8, and the conventional vaccine strains, which is genotype 2. So next. What we see here is that in the genome, there is this F protein, which is the fusion protein. There's a cleavage site there, which is very important for virulence. Next. And if you look at the sequences, if we align all the various genomes or the various strains of Newcastle disease virus, we found that there is a region of homology at the F protein. And you can see here from we call it the F cleavage site, because this site has to be cleaved by various proteases for the virus to be active. There are 3 types of petrotypes in the virus. There is the very virulent, which is known as velogenic. There is mild virulent, which is mesogenic, and that's avirulent virus, which is lentogenic. And through the sequencing, we found that the highly virulent viruses, they have got polybasic motif, and that's cleaved by intracellular proteases ubiquitously found throughout the body. While the avirulent virus, they have a monobasic, you can see here, only arginine and lysine there, monobasic motif, and it's cleaved by just extracellular proteases. Next. So what we know that the virulence marker is at the F cleavage site, and we wanted to manipulate and change from the virulent strain that we have into an avirulent strain. Next. So we did a genetic modification and created a genotype matched vaccine capable of reducing virus shedding following genotype 2 challenge. Next. To do this, we use a technique that's called reverse genetics. I think it's so common now that you can even see how you can use reverse genetics using YouTube. It's just like cooking, but it's more complicated than that. So what we do is we make the virus genome into an antivirus plasmid, and we transfect that genome into a cell, And we also transfect the 3 helper plasmids containing the NP, P, and L genes, which form the transcriptive complex. Having all these plasmids in the cell, they will be able to go through transcription, translation, and then they can have actually produce maturation, and we can form recombinant NDV viruses. So what we were able to do, we were able to swap the F cleavage site so that now we have from genotype 7, we can have now a virus which is avirulent. Next slide. Next slide. Kazima. Okay, so this is what we did. And we saw that this virus is avirulent, the recombinant strain. The MDT is 150 hours compared to the virulent NDV, which is just 58 hours. And the ICPI is 0. Okay, so it shows that the recombinant NDV is avirulent and lentogenic. Next slide. The survival for the chicken as well, we were able to show that the recombinant NDV were able to survive. Were able to infect the chicken, and the chicken was able to survive, while the wild type NDV can actually kill the chicken within a few days. Next. And that recombinant virus was also very stable genetically. Even after 5 passages of the virus, we were still able to show that there is no change in the sequence at the F cleavage site. Next. We were also able to show that the antibody titers of the chicken vaccinated with Lasota, which is the avirulent vaccine strain, and our recombinant strains about the same. Next. And also, I didn't show here, but we were able to show that the virus vaccine, which is Lasota, and our recombinant NDV completely protected against NDV, against clinical signs and mortality. The recombinant NDV significantly reduced the duration and load of cloacal virus shedding compared to the Lasota. So this is even better. And the RNDV significantly reduced the duration and load of oropharyngeal virus shedding compared to Lasota. Next slide. So this is the first genotype 7 NDV to be rescued by reverse genetics in Malaysia. And we were able to show that we were able to stably attenuate that velogenic virus. And it is the first genotype matched live attenuate vaccine candidate. And we are currently manufacturing this virus to be distributed worldwide. Next. But the journey continues. The virus can, it infects chickens and it kills chickens. But this virus can also be used as an anti-cancer agent. Next slide. This is a bit old here, the slide, but there are more than 18 million new cases of cancer cases a year, and over 9 million deaths annually. And there are more than 44 million people living with cancer. You can see here there are various types of cancers. And my interest is actually colorectal cancer. Okay, next. Now, many of you, if you look at amongst yourself in the room now, I'm sure that all of you would know someone who has been ill with cancer or who has passed away with cancer. Am I right? Now, it is such a disease. I mean, I lost my husband due to prostate cancer 2 years ago. mostly we can treat cancer by doing surgery, and then we can also go ahead and proceed with chemotherapy and radiotherapy. There are many types of cancers. They will depend on the various stages of cancers for us to follow the treatment. However, now there are newer modalities such as immunotherapy, targeted therapy, hormonal therapy, and so forth. And what I'm doing is called virotherapy. Next slide. So NDV can be used as in oncovirotherapy. This was discovered in the 1990s when they found that it is highly oncolytic and is able to selectively target and destroy various cancer cells. The advantage of using NDV is that it is very safe and generally does not cause disease in humans because it's a chicken virus. And it's also genetically stable and has immunostimulatory properties. So we did continue our research from animal vaccine to working on NDV for cancer. Next slide. Unlike healthy cells, the tumor cells have impaired interferon response, which makes them highly susceptible to NDV replication. This selectively kills cancer cells without harming the healthy ones. Now, this is so much better than chemotherapy, which is just like a time bomb in our body. Now, it kills cancer or tumor cells by various mechanisms like apoptosis, pyroptosis, necroptosis, and autophagy. Moreover, it has the capacity to activate immune cells and trigger tumor antigen release. So the tumor can also, the NDV can also spread through the giant syncytial cells. Okay, next. We know that NDV kills the cancer cells through direct and indirect pathways. In the direct pathway, we have the intrinsic pathway, extrinsic pathway, the ER stress pathway, and the RTK pathway, and many more. And the indirect pathway, we have the adaptive immunogenicity and the innate immunogenic response. Can we have the next slide? This diagram shows you basically how the virus can kill cancer cells. You can see here that in normal cells, the virus hardly infects the cells because there are no receptors for the virus and it fails to replicate. So the normal cells are not harmed. However, in cancer cells, there are many receptors because the virus attached to these receptors, which are silicic acid receptors, and they can successfully infect and replicate the cancer cells. And like all viruses that you know, they will be able to, while increasing their progenies, they will be able to lyse those cells. Next slide. There are many studies done using NDV in cancer therapy. And there's also one here. Here, this slide shows you that they've been used in I'm looking only at colorectal cancer here, and already been used in phase 1, 2, and 3 clinical trials, and with quite positive response. Next. But you can see here the numbers are small, okay? Next slide. This is an old slide, but I thought it shows clearly from this Kaplan-Meier plot that you have better survival using NDV therapy. This is overall post-operative survival of breast cancer patients. Without therapy, it's the graph is a bit worse here. Next. In another clinical trial, and this is on phase 1 and 2 trial of intravenous NDV-HUJ oncolytic virus in glioblastoma multiforme. And you can see that the tumor actually shrunk after 25 to 30 weeks of treatment starting from the virotherapy. Next. Similarly, this is on response in a man with tonsillar carcinoma. After 3 months, there seemed to be complete resolution of the tumor. Next. So we do have promising results with potential benefits, yeah, because if we can have NDV, it's a very cheap way of treating cancer. We can have improved quality of life. And also it has long-term benefits, like maybe it can provide, it can enhance the immune memory and provide long-term protection against recurrence of the cancer. And this is hard to believe, but the virus seems to attack various types of cancer cells. The only thing is I couldn't do all, so I'm only focusing on colorectal cancer cells. Next. However, there are a few limitations using the virus in oncovirotherapy. From those earlier slides, you can see that there is lack of standardized studies, and the study seems to be small, only less than 50 patients, although there was one which is over 500 patients. And there seem to be broad spectrum of clinical responses and oncolytic activity. I noticed that some of the virus strains will attack certain cancer cells, but not others. And there don't seem to be any specific pattern at the moment. And there's also presence of persistency of infection. We don't really know much about the cancer biology still. And sometimes you ask the questions, how do we facilitate the systemic spread of the virus whilst maintaining or bolstering the immune system? You have to understand that the virus itself is a pathogen, and when it enters the body wants to clear it up. Yeah, but we need to make sure that the virus is able to be in the body for a period where it can actually kill the cancer cells. We're trying to address some of these issues in our lab. Next. So using my very virulogenic or virulent NDV, we have been looking at various types of cancers, and those we use cells to look at these. Some seems to have some effect and some not so good. Next. So we found that the virus also shows persistency of infection. That is, it resists oncolysis. You can see here that the sensitive cells, before infection and after infection, the cells have died. However, the resistant cells still appear after infection. And this is what we worry about, is that we don't want to have the virus causing persistent infection, and then suddenly you have like a real-time bomb where there's a lot of viruses in the body. And also, it doesn't affect the cancer, so the cancer cells will grow. Next. When we look at the persistent NDV infection cells, we found that we can generate heat map dendrograms, identifying genes which are able to differentiate the persistently infected cells from the sensitive cells. So we were able to see that some genes are upregulated or downregulated, it depends. And we were able to see a pattern of these genes. So we are working on these at the moment. Next. How do we overcome such problems? Next. Is we try to do some little bit of genetic engineering. Just like the vaccine for the chicken, we are also doing for the human. Next. So what we do is we try to modify and introduce interleukin 12, for instance, and other immunomodulatory genes. And also we try on and modify various parts of the gene to make it more effective. Next. In here, we can see that this is an in vivo mice study. And you can see that there are, in the negative control, there are lots of mitotic bodies, but in the recombinant virus infected cells, we don't have many. Next. This is also where we have xenografted the tumor into the mouse, and we found that we were able to reduce or shrink the tumor after treatment with the recombinant virus. Next. So there is a general consensus from the clinical trials. Next. That the NDV-based therapy were generally well tolerated and demonstrated acceptable safety profiles. And the side effects observed were typically mild and transient, including those who got infected, they just show very mild flu-like symptoms and slight fever. Next. NDV has shown promising anti-tumor activity in preclinical studies and early phase clinical trials. Clinical evidence suggests that NDV can selectively target and replicate within tumor cells, leading to their destruction. Some studies have reported tumor regression, stabilization, or reduced tumor growth rates in humans, in patients treated with NDV. Next. It's also been shown to stimulate the immune system, leading to enhanced anti-tumor immune responses, and demonstrated an increase of immune cell activation, cytokine production, and tumor-specific immune responses. Next. It has also been investigated in combination with other cancer treatments, such as chemotherapy, radiation therapy, and immune checkpoint inhibitors. Some studies have reported synergistic effects when NDV was used in combination with other therapies, and this has got very strong potential in treating cancer. Next. But we do have challenges. Many people and animals are already exposed to NDV or vaccinated against it. So this can impair the efficacy of NDV-based therapies and lead to neutralization of the virus. Then production methods of the NDV need to be consistent, and we are looking at it now. There's also regulatory considerations for NDV-based therapies. We are working on it. Next. So for future directions, it does have a potential for combination therapies, and the status of ongoing clinical trials is still not that clear because there's no large study done, and we need to work on this. Next. Sorry, I like to end my talk saying that research is to see what everybody else has seen and thinking what nobody else has thought. Next. These are some of my collaborators and students that have worked with me all over the years. Next. And I like to thank all of them. Next. I think I finished.
Yes.
Now we can have questions.
Thank you very much, Madam, for very nice, comprehensive, and very well structured presentation. Now I would request our participants to please post question and answers for Professor Dr. Khadija Yusuf. Okay, so there is one question from Mr. Anayat Ali. He's asking how innate NDV kill the cancer cells in direct way.
Well, the thing is, we need to have the NDV virus has to be virulent. We do have, I think, examples of the avirulent NDV to kill cancer cells. But I think what I have tried, the vaccine strain that I used to make the vaccine against chicken, that is lantogenic, is avirulent, and I found that it doesn't have much effect on the cancer cells. So I still believe that we need to have a slightly or mildly virulent NDV for the treatment of cancer. Nevertheless, we have problem here is because the mildly virulent virus is also very virulent to our chicken industry. We have a huge poultry industry. I'm sure in Pakistan as well, you do have a huge poultry industry, and we don't want this virus to spread to our poultry and destroy our poultry industry. That's main problem that I have and I'm facing at the moment. Yeah.
Right. So, there is another question from Mr. Taseed Ahmed. He's asking your opinion that what do you see in future? What do you see is the future of virotherapy with variations in viruses across the globe, an agent infecting more than 236 species of the birds? So
well,
basically,
yeah, and hi Tanvir. The thing is, we can use the virus is safe, except for the birds. It's very safe with us humans. So what I feel is that it has got a strong, very good potential. The first virus vaccine for cancer was the Lygic by Amgen. It was passed by FDA in 2015. Yeah. For skin cancer, and it promised to be the wonder drug, but I think the sales were not as what we expected. So, but there are lots of viruses, oncolytic viruses that are being used now and focusing on cancer. So I feel that we do have a chance to see something in the near future, and I'm hoping that, and you all please make dua as well, that my NDV virus can be used for cancer treatment soon, because I'm aging and I can't wait anymore.
inshallah,
I hope that answers your questions. The rest, you can email me and we can have a private conversation later.
Right, madam. Just thank you very much, madam, again for your time. And if I would request His Excellency Professor Dr. Muhammad Iqbal Chaudhry to say some closing words if he's here with us online and has time.
Thank you very much, Sister Khadija, Professor Khadija, for a very nice presentation. Certainly a very innovative approach. Of course, viruses have been used for different kind of therapies, but using viruses and converting them into successful vaccine is a very innovative approach, and certainly deserve a lot of attention. It is scientifically significant as well as extremely interesting in terms of its health implication. So let me express my gratitude to you for sparing time for this lecture, and also to wish you all the best for your research. We look forward to have you in Pakistan, particularly in Comstech, for your presentation. And we would like you and your institution to become part of the network of Comstech, so we can work together for the benefit of Muslim Ummah. Assalamu alaikum wa rahmatullahi wa barakatuh.
Walaikum salam. It has been a pleasure giving this talk. I hope it's informative. I did cut short a bit because I didn't want to go too long. I'm having this terrible cough. I've also given in the chat box my email address. If I don't reply, just email me again and I'll try and reply. It's been a pleasure and I hope to see you all again. Do come to Malaysia and I'll try to go Pakistan. I have been there twice now. So last year and this year, it's amazing. Yeah. In fact,
Actually,
3 times. Yeah.
Yeah, we're actually thinking of sending you to the other Muslim countries also as comstech distinguished scholar. And if you can spare some time, that would benefit our brothers and sisters in other part of the world, inshallah.
Inshallah. Okay.
Thank you so much.
Well,
Assalamu alaikum.
Thank you very much, madam.
Yes.
And you can see that our participants have posted a lot of compliments for your presentation and they want to connect with you. Again, thank you very much for your time. And I would also like to thank our participants for joining in, for sparing their time to be here with us. Thank you very much, ladies and gentlemen. Assalamu alaikum and bye-bye.
Thank you, Kazima. I mean, yeah, I told you I believe in Murphy's Law. So I gave you the slides and that saved this lecture.
Yeah, so
Thank you.
we already had safeguard. Okay, thank you very much. Bye-bye.
Yeah, and you can pass the slides to anyone. It's okay. All right.
Okay, great. Thank you very much, madam. Thank you. Bye-bye.
Assalamu alaikum.